This is video 1303, and this corresponds with section 13.3 in your textbook, and we'll be talking about mutations. So first of all, what is a mutation? Well, it's a change in the genetic information, and this change can be a minor change, it could be a harmful change, it could be a lethal change. The two types of mutations we're going to talk about are gene mutations and chromosomal mutations. So in a gene mutation, this one type would be point mutations, and these usually occur at a single point in the DNA sequence. It usually occurs during replication where it's not replicated correctly, and it can involve one or more bases. Usually if it is more than one base, they're right next to each other. And now if a gene in one cell is altered, the alteration can be passed on to every cell that develops from the original one. Examples of point mutations are substitutions, insertions, and deletions. With a substitution mutation, usually one base is changed to a different base, and the effect is that one of the amino acids might be different. So if you look here, this C was changed to a guanine, to a thymine. In turn, when it replicates, now instead of having CGU for our codon, we have CAU, and that in turn changed the arginine to a histidine. Now, if say that one wasn't the one that was replaced, say it was the last base in the codon from a CGU to a CGA. So instead of the CGU, say it was CGA, and if you look at the genetic code, it would still specify the amino acid arginine. So a lot of times it's better if the substitution occurs at the last base of the codon, because a lot of times you have like UGG, UGA, if you remember. Well, those are the stop codons, but if you remember, or if you look at the genetic code, that's usually where it changes, is the last, or it doesn't change rather, if the last base is different on the codon. Now, with insertions and deletions, they're basically opposites. Insertions is where you have a nucleotide that is added, and then deletion is where you have one is deleted. So in this case, the bases are still read in groups of 3, but now what has ended up happening is that you have a frame shift, or a shift in every codon that follows the mutation. So what would be more detrimental, do you think? A frame shift mutation, which would be an insertion or a deletion, or a point mutation? Right, the frame shift mutations, because they change all the amino acids after that first mutation. So if you remember from the last one, we only changed one amino acid. This one ended up changing all 3 of them. Now with chromosomal mutations, this can change the location of genes on a chromosome, and it can even change the number of copies of some genes. So there's 4 types of chromosomal mutations, and this is when the whole chromosome is affected. These right here would be a whole chromosome. Okay? So you could have a deletion, or part of the chromosomes missing. You could have a duplication, inversion, or translocation. So the deletion involves the loss of all or part of a chromosome. So if you look at this one here where the deletion occurred, the chromosome's a lot shorter, and the section where section B is what is missing, what was deleted. In a duplication chromosome, chromosomal mutation, usually the resulting chromosome is larger, and you have an area where it's repeated, so it was duplicated. In an inversion chromosome is where the genes are not in the correct order. So here, when A, B, C, D, E, F, now we have A, C, B, D, E, F. And what you're going to need to be able to do is be able to look at the original and then look at the mutated chromosome and be able to say what type of chromosome or mutation has occurred. Now translocation occurs when a part of one chromosome breaks off and attaches to a different chromosome. So that's our translocation. So in this case, we have GH, which broke off from some chromosome, attached to the ABCDEF chromosome, and it attached in between the B and the C gene. So what's the effect of mutations? First, you could have little or no effect. We talked about that in class, just depending on where it is. It could produce a beneficial variation, or it could be very negative and disrupt the gene function, period. Mutations often produce proteins with new or altered functions that can be useful to organisms in different or changing environments. In this picture here, we've got a two-headed turtle, which is a mutation. Do you know what the scientific word would be for an organism that has two heads? Do you remember what the prefix is for two? And then what's the prefix for head? You should know these now. What can cause mutations? So earlier in the year we talked about mutagens. Remember, we talked about smoking, x-rays. Was smoking, x-rays, asbestos, the sun, UV rays. But what also can cause mutations are errors during replication, okay, environmental conditions, and the mutagens, like I just mentioned. Here, showing you, I gave you a picture of flies, fruit flies, and we had a normal male and a normal female, and then their offspring was a mutant. And if you look, it's kind of like a mixture of the two. The eyes are a different color. The abdominal area is a little bit different. Okay. That's it.